here is really to talk about the supporting structures of the central nervous system, the physical supporting structures. So we've got the bone, which is the skull. This is really the cranium or the skull cavity itself, but also think the vertebral column as well, because it protects the spinal cord. We've also got the meninges. Again, we talked at length about the meninges in lab, so I'm not going to go into too much detail here. But the meninges have protective ability as well. There are 3 layers that we talked about: dura mater, arachnoid mater, which has the blood vessels, the cerebrospinal fluid within that layer. And then we've got the pia mater, which is the more intimate layer that's connected to the brain and spinal cord. So I got a question here about whether or not our bodies have a fixed number of neurons. And we kind of pretty much do now. The number of neurons that we create, so during the initial period of development, during infancy, anywhere up to age 5, 6 or so, we're forming lots of new neural connections, lots of synapses are being made. And then beyond that stage of life, we really don't see a lot of variability in the neuron production because neurons cannot replicate themselves, and we don't generally produce new neurons beyond that period. What we do are we make or modulate or change existing neural connections. So we can make new synaptic connections between the existing neurons, but we don't usually add new neurons beyond that developmental period. Okay, excellent question. All right, so just kind of an overview of the structures here again. So again, we've got the skull, we've got the meninges, as well as the CSF that's floating in the subarachnoid layer. And all 3 of these structures provide support to the brain, okay? Both the skull, the meninges themselves, and the cerebrospinal fluid help to protect the brain in the skull and offer structural stability, structural support. Okay, and so let's talk a little bit about CSF here. So CSF, or cerebrospinal fluid, is created in the created by the ependymal cells. It is circulating in the ventricles. So we talked at length about the ventricular system, the lateral ventricles, third ventricle, fourth ventricle, and it's this clear watery fluid that is bathing the central nervous system, and it's also helping to suspend the brain. So we talked about the fact that because it's in the subarachnoid layer, one of the layers of the meninges, it's really suspending the brain and preventing the brain from sloshing around or hitting against the tough cranial bone and injuring that delicate brain tissue. So it is secreted by the ependymal cells, one of our glial cells that we talked about earlier. These are the cells that line the choroid plexus. So if you remember the choroid plexus again from lab, this is where CSF is produced. It then circulates through the ventricles, and then it's finally reabsorbed back into the venous system through the arachnoid villi. So if we jump back to this slide, we can see these villi. The villi are basically these outpouchings or outpockets, these granulations. They provide communication from the subarachnoid space where we have the CSF floating to the sinuses, and the sinuses communicate directly into the venous system. And so this is how CSF can be secreted and then reabsorbed, secreted, and then reabsorbed cyclically in order to continually provide new CSF and to continually circulate that CSF around the brain and spinal cord. And so again, it cushions the brain as well as maintains a stable interstitial fluid environment. So it's one way to prevent the brain tissue from being exposed to the interstitial fluid that's bathing other parts of the body and from the environment in the plasma as well. So there's a distinct, unique fluid environment that's circulated around our brain. And if we look at the actual composition of CSF and we compare it to plasma, it's pretty much the same with slight variability, but I do want to point out the absence of proteins. And we'll talk about the blood-brain barrier, and proteins are really large. They can't actually get through that barrier easily. And so we'll see this large difference between the quantity of proteins in the plasma compared to the quantity of proteins in CSF. As far as other substances, our other ions and electrolytes, they're pretty much the same between plasma and CSF. Here we're just reviewing the ventricular system. Again, I'm just going to point these out briefly, but we talked about these in lab at length. So we've got the lateral ventricles, we've got third ventricle, we've got fourth ventricle, and the various communicating structures. And this is really where CSF is made. It begins to circulate, and then it goes all the way around the meninges that cover the spinal cord as well. So it's not only circulating around the brain, but it's also circulating around the spinal cord as well. All right, let's move on to lecture objective 3. This is talking about the significance of the blood-brain barrier. And so the blood-brain barrier is what helps to distinguish between the nervous tissue, as I mentioned, and what's happening in the plasma or in the blood. So the brain is really small in terms of its weight when compared to the rest of the body. It's about 2% of our total body weight, but it receives a large chunk of our cardiac output. It receives about 15% of the blood supply that's circulated throughout the body. So that's a lot of blood when compared to the size and weight of the brain itself. And that's because it has a high metabolic rate. It requires a lot of oxygen, even at rest, and a lot of glucose, again, even at rest, when compared to other organ systems and other structures. And that's really because the brain is kind of always on, right? Even when we're sleeping and resting, the brain is always regulating other body functions, creating memories, imprinting short-term memories to long-term memories. There's a lot happening in the brain, again, that we still don't quite understand, like dreams and all that type of activity that happens even while we're sleeping. And so it's heavily energetically dependent. It requires lots of oxygen, lots of glucose. And we'll see that if there's any interruption of glucose supply or oxygen supply, the consequences can be quite detrimental and permanent to the brain tissue. So neurons rely on aerobic glycolysis. This is production of energy in the presence of oxygen. It's going to require a steady supply of glucose and oxygen. As I mentioned, even a slight interruption in the oxygen of the brain, even 3 to 5 minutes, can produce permanent brain damage. If you think about cerebral palsy, I'm sure many of you have heard about cerebral palsy. This is when a fetus is coming through the birth canal. It's a very sensitive time. If there's interruption in oxygenation, even for the slightest second there can be long-term permanent consequences from neurocognitive dysfunction all the way to full movement disorders from that interruption in oxygenation. Now, again, the brain doesn't have the ability to store glycogen, and so the glial cells have some capability to synthesize glycogen, and the brain relies heavily on glucose, a steady supply of glucose. It cannot rely on fatty acid oxidate or beta oxidation from fatty acids as easily. Other tissues have the ability to do that, to break down fatty acids and produce ketones as a way to provide an alternate source of energy, and the brain doesn't have the ability to do that very well. So it's very heavily reliant on a steady supply of glucose or glycogen. Now, whenever there's an interruption in blood supply to the brain, this is called a stroke. There are two types of strokes. There can be an occlusive stroke, which is where there's an occlusion or a blockage in a blood vessel. There can also be a hemorrhagic stroke, where there's an actual bleed or an injury to a blood vessel as well. And as you would imagine, there are lots of different presentations for strokes, from mild visual disturbances, there can be speech disturbances, movement disturbances, one side of the body, depending on what side of the brain that injury, occlusion, or bleed is found on. All right, let's move on. Again, looking here at the blood-brain barrier, it's formed by the capillaries of the central nervous system. These are joined together by very secure, tight junctions, as well as the astrocytes that we talked about before. So the blood-brain barrier is a very selective barrier that prevents the exposure of the central nervous tissue from things that are happening in the capillaries in the blood itself. And it's really formed by, again, the tight junctions between the endothelial cells of the central nervous tissue capillaries, or the central nervous system capillaries, as well as the existence of the astrocytes. and the foot processes of these astrocytes, right? Remember these star-shaped cells that have these foot projections or foot processes that also help to secure the communication between the blood and the nervous tissue or the neurons that are in the central nervous system. So it really helps to prevent the movement of large molecules, again, of unwanted molecules. So as we can see from the capillaries here, proteins cannot escape easily. They're too large to get through. We do have some select movement of hydrophilic substances that can go through the membrane via transport, and as well as hydrophobic substances that can go right through the membrane because they can go through that lipid bilayer. But otherwise, there isn't much movement across this barrier. And again, this is a way to protect the brain tissue, to protect the neurons from any type of injurious exposure from substances that are traveling in the capillaries or in the blood. And then objective 4 here talks about myelination, so how we myelinate the nervous system. We talked at length about why myelin is important, right? The speeding up of the conduction velocity of neurons, as well as the nodes of Ranvier, those gaps in between the myelin. So now we'll talk about the cells that actually produce the myelin. And myelin is produced differently in the central nervous system when compared to the peripheral nervous system. And so we'll start by looking at the myelination in the central nervous system, which happens with the oligodendrocytes. So oligodendrocytes are the cells, they're part of the glial or the neuroglia collection of cells. They myelinate the brain and spinal cord specifically. They pretty much send their axonic projections or their extensions, excuse me, to cover many axons at a time and basically myelinate those tissues respectively. So we can see one oligodendrocyte here, and we can kind of point it out by its nucleus. And then it sends these different projections to many different neurons, many different axons, and it myelinates those axons, again, providing those gaps that we talked about, which are the nodes of Ranvier. Now, myelin is what gives the brain tissue its white appearance. This is how we distinguish between white matter and gray matter. So white matter is really the fatty tissue that's really laid down as this myelination. And this is really the axons of the central nervous system. This is compared or contrasted to gray matter. And gray matter is really the somas or the cell bodies and the dendrites. And these are not myelinated, and so they don't appear white, and so we call them gray matter. So white matter is the presence of myelin. It's usually the axons of the central nervous system or the neurons of the central nervous system, whereas gray matter are the proximal parts of the neuron, so the soma, the dendrites, and this area lacks myelin. That's why we call it gray. And this is usually, again, within this, the actual soma of the neurons themselves. Now we can speak about Neurilemma. Actually, let's go back to this slide here quickly. So I want to mention that oligodendrocytes myelinate, but they don't associate with Neurilemma. And I'll talk about what Neurilemma is as it relates to the peripheral nervous system myelination. So Neurilemma is basically the wrapping of the Schwann cell. The Schwann cell is the cell that myelinates outside of the central nervous system. So pretty much everything excluding brain and spinal cord, the peripheral nervous system. And the Schwann cells wrap themselves around the neuron and they provide this Neurilemma. Now, Neurilemma is not the myelination itself, but the Neurilemma is what's used to provide or produce the myelination. So let's look at this neuron. So here's a peripheral nervous system neuron. Here we can see the Schwann cell itself, the nucleus of the Schwann cell. And we can also see that it begins to provide this wrapping around, this sheath-like projection that wraps around the axon of the neuron. So the blue here that's shown is the actual axon of the neuron, and the layers that are being wrapped around are the production, the sheaths that are produced, that's called the Neurilemma. And so it wraps around, again, providing the gaps, which we call those nodes of Ranvier, and myelinates in the peripheral nervous system tissue. Now, as we know, the smaller neurons do not have myelination, but they do have neurilemma. They do still have an association with Schwann cells. So neurilemma is not the myelin itself. We can have an unmyelinated neuron that's still associated with neurilemma, still associated with Schwann cells. And we see this with our smaller axons, right, that are not myelinated, that do not require that speeding up of the action potential velocity, as well as some of the sensory systems, the autonomic nervous system. We also see some of these smaller unmyelinated neurons, but they are still associated with the wrapping, that sheath-like wrapping of the Schwann cell, which is called the neurilemma. All right. So before we kind of look at these checkpoint questions, I'm going to pause again and see what questions you may have.
This is from the last, it was from like the beginning of lecture, but could you explain what phagocytosis is again? Just like what it encompasses?
Yep, sure. So I'm going to jump back. This was one of our glial cells, specifically the microglia cells. And the microglia cells are the immune cells of the brain. So phagocytosis is the ability to engulf toxins, dead or injured cells, cellular debris, bacteria, or viruses. And so it's really a function of the immune system, and it's the ability to kind of wrap around that material, whatever it is, bacteria or a dead or dying or injured cell, and break it down into smaller pieces and discard of it. And that's what phagocytosis is, and that's what the microglia cells do. Now, phagocytosis is a function of other cells. We'll talk about macrophages and neutrophils that have the ability to phagocytose in other parts of the body, but specifically in the nervous system, phagocytosis is done by the microglia cells, and it's a way to remove toxins or, harmful exposure, harmful substances from the nervous tissue. Okay. Okay, excellent. So someone's asking to go over Schwann cells. So Schwann cells, we said, are the cells that myelinate specifically in the peripheral nervous system. They're associated with the neurilemma that I talked about. So the neurilemma is the wrapping of the Schwann cell that forms this sheath-like wrapping around the axon, and then it produces this myelination, this fatty myelin layer that provides the myelination to the cell. Now, again, it's producing these gaps along the way that allows the speeding up or the increasing in the velocity of conduction of the action potential on these neurons. So that's the role of Schwann cells. And we want to contrast Schwann cells from oligodendrocytes. Oligodendrocytes myelinate in the central nervous system. They are not associated with neurilemma, and they usually have the ability to wrap their extensions around many different axons and myelinate those axons as well. So it's just basically the difference in central nervous system versus peripheral nervous system and the association with neurilemma, with which the Schwann cells have and the oligodendrocytes do not.